Thank you, Nissan, for your kindly introduction and very good reason, very convincible to tell everyone the importance of a PhD. But I thought today I'd like to share some of our work and tell you the importance of them. And at the end, I would like to share some information about my department. So currently, I work in the Department of Medicinal and Applied Chemistry in Kaohsiung Medical University. I serve as a chairman at this moment. Okay, so my research topic is about the synthesis of peptide and dendrimer for precision medicine. Okay, all right. So this is a probably very rough blueprint about my research, and my work is based on the solid phase. So if you are a chemist, I can provide you a lot of interesting work. So we use a solid phase synthesis to prepare dendrimer and peptide, and then we use the product for all different kind of purpose. But because of my personal interest and the preference of my university, most of the work is related to the biomedical application, like antibiotic, stroke, treatment of stroke, and the pet aging, that kind of job. Okay. So I believe most of us understand the importance of the narrow medicine. So the narrow medicine means you use a compound in a narrow scale. So they have a very unique property, and they've been developed for more than several decades. And until now, a very unique and very beautiful property have been discovered. However, there is still some problem. So although there's around 80 market product on the market, okay, so they still have some problem. And among all the narrow material, we work on the dendrimer. So one of the beauty of the dendrimer is they are very unique and very well-defined structure. So we have a lot of branch side. So compared to other polymer, so we have very dense distribution of the surface. So at the end, we can produce a very pure compound for the biomedical application. And if they've been developed from the first compound being launched around 1987. So since 4 decades, they penetrate all different field of application. So this is one paper published in Science Translational Medicine this year. So even the dendrimer alone can promote the therapeutic efficiency to the COVID-19 patient. So you can see that it already get into a phase 2A clinical trial. So the challenge of all the dendrimer and even all the narrow medicine is mass production and the purity and their monodispersity. So that means that you need to get a pure compound. So they have all different kind of application. I'd like to mention that again. But these two is the major problem. And since their compound have very big mass, so they don't have any good way to solve this issue. So for this problem, we start this job probably 10 years ago. So we figure out a solid phase is a very good and a very simple way, since the solid phase synthesis have already worked for the peptide, even for the therapeutic peptide for more than, I think, 20 to 30 years. And their size is pretty close to the narrow medicine level. So to make a long story short, so instead of a linear monomer, so we make a branch monomer and they use a solid support. So apply a similar job. But of course, the different skill, okay, the idea is similar. So you repeating, introduce your branch monomer. So eventually you can build a compound with a very big one. So that is the dendrimer. You have a branch side, you see A, B, C. So you have a 2 branch. So gradually you increase the branch number of your surface group. Okay, so that is the point. So this is one of the examples. So we can build a compound to like a very big one. The molecular weight is, I think the molecular weight is around 50,000 with a very pure product. That have never been done before. So that is a very important skill and a very big leap in this field. So later we use this skill to prepare a certain compound for different application. And one of the application is to synthesize the amphiphilic compound. So recently the assembling of the compound to be a narrow vessel or a vehicle is very attractive topic. So you can encapture like RNA and with a targeting moiety and get into cell. So as you may know, even the Moderna's vaccine against the COVID-19 use a similar skill, but they use the liposome to encapture the RNA. But instead, we use a branch compound on the surface. So with this type of compound, we can maintain the dense distribution of the surface, create a more diverse and a more attractive application than the regular liposome. However, this type of compound is very difficult to synthesize and application. So don't forget for biomedical, the purity of your compound, no matter in any part of your product, is very, very important. So you still hear me? I saw
yes. We can hear you.
I saw a warning that's unstable internet. Okay, so that's why I asked that.
Yes.
Okay, so by our solid phase work, we divided that into two work. So again, make a long story short. So you can see we can produce all different kind of even more diverse compound with a aliphatic chain that is hydrophobic part and very much the hydrophilic surface and even with all different kind of modification. Nowadays, we can produce more diverse compound. I can show you a very. Sorry.
No, sorry. Go ahead.
Okay. So I will show you later about the application. So the other part of our work is on the C-terminal modification of the peptide. So this is a very quick scheme to tell you about the solid phase of the peptide. I believe some of you may learn this part in the undergraduate chemistry courses. Okay, here is a resin. So you incorporate a peptide here and then remove the protecting group and get into your product. And the deprotect, you make it a circle. So you're repeating the deprotection and the coupling, so you can make your compound very long. And then cleavage from here, so you can get your product. So this compound being developed by the Merrifield from 1962. Until now, the skill and the technique is still follows the similar way. However, there's one problem. This part is this part. So this part been attached to the resin. So it's very difficult to functionize this part of the peptide. However, a lot of biomedical product need that. So for this one, for this purpose, we design a new resin. So here is a new resin, okay, being developed by Indian student, Dr. Anna. So we use this one. We can introduce all different type of nucleophile, small one like a small molecule, or even Dendrimer, another peptide. Or nowadays we even use this skill to functionize a narrow particle. So we can create a functional narrow particle. So this is one example. You see, we can produce a very short peptide and use another part of the peptide to attack. So you can get a dimer and then trimer the same way. You use this one to attack this position, so you can get trimer. The similar skill will give you a tetramer. So you see, we instead of incorporate peptide by one amino acid, we make the skill become a assembling part. So you can divide that into 5 mer and put them together. And that is also a very important skill nowadays in the vaccine development. So we have a collaboration talk about this part. Okay, that is a synthesis. So later I will show you a very quick tour about the two of the biological study in my group. One is a blood-brain barrier transporter. Okay, so to find out the compound can be suitable for the blood-brain barrier transporter. So we use this skill. So that is a chamber that we incubate cell here. So we put the compound on the up layer. So from after like 2 to 3 hours, we analyze the compound in these two parts. So here, that is the result. So we can understand each compound, their ability to penetrate a cell. Okay, so if we use the brain endothelial cell, so we can simulate the result of the blood-brain barrier. So that is very important in the therapy of brain disease. So in this project, we focus on the glioma, that is a brain tumor. Okay, so we synthesize all different kind of peptides. So based on the skill I just show you. So here is one example. You can see from the chamber. So let me back from the in vitro model, we find the compound and then use those compound, those peptide with a fluorescent style. So you can take we can do the animal study and take those like this iris image. So you can see the compound really get into brain. So that means the model is really work. So we're still working on this part and collaborate with several Europe group to develop a vehicle for against the brain tumors. The other part is anti-biofilm material. So as you can see, that is under that is a ship. The part of the underwater, you can see a lot of seashell on the surface. So this is very quick.
Professor, can you wait a little bit? Sakarika, can you check who has the noise, please?
Okay.
Yes, Lin, I'm checking, Lin.
Yes, sorry. Yeah, because that...
No, it's fine.
Yeah. Okay. Thank you. Go ahead.
Go ahead now.
Yes. Go ahead.
Okay. So that is a very quick tool about the seashell formation on the underwater material. Okay, not even under the ship. Nowadays, the gas pipeline and the underwater cable have a similar problem. So the first one is you have to attach a cell and it grows a biofilm, and then the second bacteria attach, and then grow more complicated microbiota, and then grows another sea plant and any other material, and then grows a seashell, and then they cover the whole material and cause a lot of trouble. So nowadays, we like to produce a material for this purpose. So there's several ways that people use for this against the falling problem. So quickly tell you, the very old one is a toxic material, like you plant a lead or mercury on the material, but that is not good because that will kill the marine animal. The other one is you use the antibacterial agent, so that is stop the first step of the bacteria attach, but that's also cause problem. And the other one is called painting the hydrophobic material like Teflon. But that is also cause another trouble. The reason is Teflon, the attachment of Teflon is not very good. So this causes a toxic and accumulation of plastic in seawater. So the latest one is use a liquid iron material. So the idea is showing some in this figure. So you have a material plain on the you have a coating but plain on the material. They have both a negative charge and a positive charge. So they can prevent those type of cell attachment. So they cannot attach the cell. So nowadays we use our peptide skill to synthesize those peptide with liquid iron and use in this purpose. But sorry for the IP issue that I cannot show you the detail, but we do all the anti-biofilm experiment in my lab, and of course the synthesis of the material, okay? And for the work I just told you that we have two collaboration with Europe, that is a Euro narrow-matter research project. If you are interested, you can look at these two YouTube file. I believe that the file will live in EduCare Taiwan, so you probably can see that detail. So that is a two different projects with Europe, and all the project being supported by our government and the Euro narrow-matter project office, and of course, and the other of course our university and the Embassy of French in Taiwan.